Hey everyone, how's it going?
Hi guys.
So we'll maybe wait just a bit longer if anyone else wants to join, and then we'll get started. How was the first 3 days of medicine been? Pranay, do you want to answer?
Yeah, it's been pretty good.
Pretty
A lot
good.
of work.
A lot of work. Okay.
Yeah, kind of just
Um,
big
yeah,
change from the holidays, so gotta get used
yeah,
to it.
yeah, for sure. Is anyone here behind on lectures? Nah? Okay. Then you've already made a good start.
And to be honest, I don't know if I'm behind or like on track. I'm a bit lost. Like, am I
Yeah,
supposed
yeah.
to study hours on each
Hey,
lecture
you're
or
okay.
like just
Yeah,
view them? I'm not sure where I'm at, you know.
yeah, no. Trust
That's
me,
what
those are
we're here for.
questions we were all asking ourselves this time last year. Yeah. I think it's all about just figuring out, what works best for you. Some people spend hours on lectures, some people don't view lectures at all. There's two ends to the spectrum. So yeah, just figure it out. I think the best way to know how you're tracking is at one stage maybe pull up some of the past exam or like exam questions, see if you can answer any of those. And the good thing about our PSP, sorry to just plug it already, but we run Kahoots and quizzes and all these, we try and give a lot of questions. So in that way, if you complete a Kahoot and you're getting like 2 out of 10, then maybe that's a sign that you might need to, figure things out a bit more. But if you're, consistently getting 10 out of 10, then I think that's a pretty good sign that what you're doing now is good.
Perfect. I was wondering, since you mentioned that I keep asking for exam-like questions to guide my studies, where can I find those?
Yeah, no, good question. So I guess it's pretty early at this stage. You're in your third day of medical school. So yeah, like you definitely we can send a link through or we can post on our Facebook page. There's this database called Aquella, something like that, where they have past exams. So you can check it out. But personally, I probably didn't look at them until like week 10, week 9 of semester. So yeah, but if you do need it, yeah, we can get that through.
Yes, that would be great. Thank you.
No
Yeah,
worries. Just looking at the chat as well. Yeah, Ashwini, do you want to take it away? Yeah.
Yeah, I was just going to quickly, before we begin, add on to what Yash is saying. A lot of medicine, we'll sort of talk to you guys about how we would recommend to study in medicine and all of that, but a lot of it is, it all sort of adds up together. So it's hard to start doing questions really early on because you won't be able to answer some parts of the question. So like Yash said, it's usually a little bit better to wait till a little bit later in the semester to start doing the questions so that you're not almost wasting the question without actually knowing the content.
Yeah. Well said, as always. And are there any other questions, Ashwini, in the chat that we need to answer before we get started? Or
Are the meetings gonna be this time every week?
yeah.
Like all over the fluctuate. Okay.
Yeah, I think we're looking. So I know that there's a couple clashes with some PSPs, but we want to be different. So we're doing it on a Wednesday. Do you guys have a day off on Wednesday, right?
Yeah.
Okay. So yeah, hopefully this works well, and let us know as well if this sort of thing suits. Yeah.
All right, can everyone see my screen?
Yes.
Yep. All right, so shall we get started? All right, cool. So as you guys know, we're PSParachute. So basically we're here to try and make your life through first year of med as easy as we possibly can. It's not an easy task to get through the first year of medicine, but we're here basically to make that as easy as we physically can. So this is the team. We have Yufei, Mahek, Yash, Lakshita, I'm Ashwini, Akanksha, and Sav. So yeah, basically we're PSParachute, and like if you guys want to contact us in any way, you can contact us through Facebook, either directly through the PSParachute page, or you can DM any one of us, and we'll be happy to answer any of your questions. So we just have a little bit of advice that we wanted to give to you guys because yeah, it's often good to have a little bit of advice starting first year med because it is tough. So let's start with Before we start with that though, I just want to go through basically, someone asked before why there are so many different PSPs. And it's basically because each PSP will run their program a little bit differently. So the way that we're planning to run it is by giving you the content for, say, week 2 before the beginning of week 2, so that you can have a chance to sort of introduce yourself to the content before the lectures, so you're not just bombarded by a bunch of information when you do watch the lectures. And then we plan to have sessions like this in the middle of the week. And at the bottom there, you can see that we've said have polls to make sessions more tailored to you. So our plan is that before we run these sessions is to put out a poll and ask which areas you want us to cover more specifically and more in detail, so that we're not just wasting your time and we're not covering content that you guys already know reasonably well. We're also going to do some Kahoots that will cover the content from the previous week, just to make sure that you guys are constantly getting that revision in, and you're not just learning the content and then immediately forgetting it. Yeah, so you'll have access to the slides and everything before the week itself, and we're also going to be putting up these recordings on YouTube, and we'll give you our YouTube link at some point. It should also be up on Facebook. Yeah, so this is the advice from the team. So Sav, if you want to start for us.
Yes, hi guys, so my name is Sav, and my advice basically is do not procrastinate. You might be accustomed to putting off important things to the last minute from high school, but I can tell you that if you do that in med school, you're going to fall behind very quickly and everything will snowball faster than you, before you know it. So yeah, just don't procrastinate. And my other advice besides that is to prepare a timetable before each semester, or well, maybe it's a bit too late for that now, but at least at the start of the semester with so go through both your unit guides and see what assessments you have for the semester, and just kind of break them down and plan out how you're going to approach them so that you can avoid cramming last minute. And my other advice, important advice, is to try watching your videos at minimum 2 times speed. I found that even watching at 6 times speed, you can learn stuff and you get it done in 10 minutes. It actually works, not gonna lie, but it depends on you, of course. I'd recommend you try it though. Okay, so let's hear from Lakshita now and see what her advice is.
Hey everybody. So my first piece of advice is the best time to try out new study techniques is right now. So whether that be you like trying questions or you like handwriting your notes or typing your notes, now's the best time to figure that out, because later on you probably won't have the time. So I suggest using that time to do that. Second thing is try revise as often as you can. Because I remember last year I used to procrastinate a lot, especially when exams were coming. So I suggest not to do that. And third thing is by focusing on what is actually important, what's going to come up in the exams and what you're actually going to be using in the future, you can be a bit more relaxed and you'll find that there's less stuff that you actually have to learn and memorize and keep in your mind. Yeah, so who's next?
Sorry, just one thing I wanted to add, by the way, sorry, to the third piece of advice I had about speeding up videos. I use a Chrome extension called Video Speed Controller, which can go all the way up to 16 times if you're really keen. So yeah.
Yep. So I think I'm next. So hey guys, once again, I'm Yash. So I've got a few pieces of advice, and they're quite similar to Lakshita, but we'll give it a go. So I think the first one really is that everyone studies medicine in a different way, and you know there's no one size fits all sort of technique to how to take lecture notes or how to learn new content. So for me, like to be completely honest, I think it took me the best part of the first 6 months of last year during lockdown to kind of figure out should I use OneNote, should I use Notability, should I use GoodNotes, should I use PSPs, all these different things. So experiment, see what gives you the best yield, and then try and stick with that. So yeah, definitely experiment and give things a crack. The second piece of advice is really about enjoying the journey and the ride of medicine. So, we get told even in year 2 that a lot of the content you'll find is quite confusing at the start. And even in year 2, they've told us that the first 6 weeks of anatomy, you'll feel really confused and nothing really will be coming together. But, my piece of advice to you is even in year 1, really embrace that uncertainty and know that, it's the uncertainty that will, make you learn better and all these different things. And I can guarantee you that, 2 weeks before the exam, everything will come together. So just don't get worried and enjoy that uncertainty as hard as it is. Another thing is that the people in medicine are really incredible. So even though we were online last year, I think a lot of us were still able to make really close friends, which is pretty much a testament to the sort of people that are in the course. So reach out, form study groups, talk to, a range of people, all that. And I think you'll get a lot from that as well. Second last point is try and stay as up to date as possible. It is really hard. You will inevitably, most people will inevitably fall behind. But yeah, just really try and stay on top of medicine. It's one of those courses where you can really easily be like 6 weeks behind, and at that stage, it's kind of impossible to catch up. So yeah, try and stay on task. And the last one as well is just live a balanced lifestyle. So still engage in sport and any other extracurriculars that you have. There's a lot of clubs at university as well, so maybe try your hand at one of those. And now Mehek.
Yeah, hi guys. Hopefully you can hear me all right. Can everyone hear me all right? Yeah. Okay, perfect. So my advice mainly, so some of them are quite similar to Yash and Lakshita, but first one is basically take some time out, even if it's just one day or at the end of the week, just to revise what you've learned. You can just draw a mind map, or you can read through your notes, whatever it takes. It really helps to revise the content and again, which is something that I learned last year. And my second advice would be to do not stress if you haven't found what study technique works for you, because like Yash and Lakshita mentioned, it takes time. It definitely takes a lot of time. I personally used to handwrite my notes, and I found that was really time consuming last year, so I had to change that up. But basically try different techniques, what works for you. Some people use an iPad to note take, which is definitely fine as well. And an example is Anki. So I know a lot of people used Anki last year, and it's basically flashcards that help you memorize and revise a bit of the content. And I personally it didn't work for me as well. So don't stress if that doesn't work as well as good for you. Like Akansha said, it does give anxiety, definitely. But yeah, and then my third advice would be to try work in groups. So I know you guys have just started off, you might not know that many people, but just as Yash said, go out of your comfort zone and try to meet new people. And that helps fill in the gaps when you're studying, because something you might not know, other people tend to know or might know. So yeah, that's a great way to study. So yeah, these are my 3 advices. On to the next person.
All right. So my pieces of advice, well, first of all, it's really important that you guys find a balance between uni and any other activities that you're doing. It really does become tempting to just feel yourself just having to study all the time. And it's really just not a healthy way to go about it. I know this for a fact. I definitely fell into this thing when I was in first year, and it's just not good for your mental health. You can't study effectively either way. You can't study for hours on end. So definitely make sure that you're like going out with friends, doing some sort of exercise, all that sort of jazz. Just try not to just sit in your room and do lectures for hours and hours. This is something that's already been touched on a little bit, like work with other people, ask each other questions, and try and re-explain the concepts that you've just learned. We've all heard that before. If you can re-explain something to someone, then that means that you've really understood it. And so, yeah, obviously that's a really good way to go about it. And the last thing that I would say is, so I know that before we were talking about questions and exam papers and all of that. If you're someone who really thrives through doing questions and that sort of learning, I would recommend creating questions for yourself and then testing yourself on those. So that can be through Anki, it can be just by writing out the questions anyway. But I think that's something that I definitely thought was really useful for me when I was in first year.
Hey guys, so my advice is pretty similar to what everyone else has said, but my first one is don't compare your study techniques to everyone else's. In medicine, it's really intimidating sometimes when you enter a tutorial and everyone seems like they know everything and people are using Anki and like these fancy techniques like active recall and spaced repetition and stuff that I personally had never even heard of before because I wasn't from Melbourne. These like weren't popular things at my school. But what's working for everyone else may not actually work for you. So like personally, I didn't really like using Anki. So yeah, just don't fall into that trap of comparing yourself to other people's study techniques because just because it's working for them and might seem like it's a really effective thing doesn't mean it's necessarily going to work for you. The next thing is to watch all your lectures before the corresponding tutorial. And that's because honestly, if you haven't watched the lecture, you may as well not attend the tutorial because tutorials almost assume that you have watched the lecture and somewhat understand it because what they try and do in tutorials is help you apply that knowledge and build on it even further. So tutorials won't always just be revising what was in the lectures, but rather giving you new information on the same topic. So it's important that you have actually at least watched the lecture, even if you don't fully understand it yet before the tutorial. And lastly, like Sav said, don't fall behind because as you fall more and more behind, things just build up even more and more. And I found that the difficulty level of the content really increased as we progressed through the semester. So for example, if you've done year 12 bio, the first few weeks will be quite approachable of semester one because a lot of biochem, like for me, was just revision. But as you move through, there'll be more and more new content. So something that I used was just following the timetable that the faculty gave to us and trying to watch all the lectures within the given time slot and things like that, just as a way of not falling behind. But I know some people make their own study timetables up as well.
All right, lucky last. Right. So basically my advice, I don't really have any like study advice because to be very honest, I didn't study that much in first year. But here I am, I'm fine. So basically my first advice is kind of related to that, which is basically figure out your priorities. So like for some people, it might be they really want to study a lot, they really want to kind of get the HD or like get really high grades and like understand everything. Then that's amazing, that's totally fine. But for some people, you might have like say other commitments or like you want to focus on other things, maybe in first year or like in med school. That's totally fine as well. So I think like kind of the advice and like your study method would really tailor to your priorities and like what you want to focus on and how you want to develop during your years in medical school. So really just like don't get that pressured by people around you and like don't get too intimidated because like there are a lot of smart people in medicine and like usually like when you say you go to a tutorial and you'll find like a lot of people just they know a lot of things and like maybe you don't know as much, but that's fine. Well, if you think it's fine, then it's fine. It's like if you think I don't have to know that much, then that's fine. Yeah, and also second advice would be get to know more people, make friends. And so for me, I'm an international student, so I didn't know anyone in Melbourne. So I think for my first semester, I didn't make that many friends either. So that was like quite a sad experience. But I do find that like after I've met some people, after I've met some friends, my experience became so much better. And it's not only just your social life, but also like your study, your like academic side is a lot better as well. So definitely like if you're feeling a bit say lonely or if you're kind of struggling to make friends, definitely like reach out to people, like reach out to maybe one of us. Yeah, we'll definitely be happy to help out. Third advice, so yeah, that's kind of similar to my first advice as well. So like again, I feel like I'm really not giving that much study advice, but yeah, it's like in first year uni, it is a great time to try out new things. So in terms of your study techniques and also other skills as well. So I think uni is really different from high school in that in high school, you might just be aiming for like one goal, which is, oh, I want to get into med, or like I want to get into this course, I want to do. But in uni, like you have like so many possibilities and like a lot of things you could explore. So definitely take that chance and go and explore those things. And yeah, a bonus advice, don't stress too much. You'll be fine. We'll all be fine. Right, that's it. I'll be handing it to Yash.
Awesome. So just a few things before I get started. I don't know if we've mentioned, but we hope to make our PSP as sort of high yield as possible. And what we mean by high yield is that we'll really be focusing on content that will be examined at the end of the day. So for me, and for probably you, Faye, as well, we'll be covering cells 1 and cells 2. And to be honest with you, there is very, very little of those 2 lectures that you'll probably see on your end of semester exam or end of year exam as well. So we'll be really, sort of going through it quick. But in the case that we do go through it like way too quickly, feel free to pop any questions into the chat as well. So yeah, let's get started. So yeah, firstly, I guess we wanted to talk about what is a cell and what are the sort of cell theories that you would have learned this week. So the two big ones are the traditional cell theory and the modern cell theory. In terms of some of the main points here, the traditional cell theory states that all cells come from other cells, which is really important. Another big sort of breakthrough was the fact that all life is composed of one or more cells. And in modern cell theory, it's that cells contain DNA, which contains the instructions for life and is passed down from generation to generation and through cellular division as well, which we will cover today. And all cells in a closely related species must function similarly to one another. And I vaguely remember that point being something that a lot of people forgot and something that they can test. So next slide. So again, yeah, I didn't do VC bio, so a lot of this last year was new. So I thought for those who also didn't do VC bio, this might be good. So the cytoplasm, so now I'll be talking about sort of the contents of a cell. Now the cytoplasm is pretty much that sort of watery liquid substance inside of a cell. And we define it as the contents of a cell minus the nucleus. Organelles are, the way I remembered it, was sort of the organs of a cell. They're specialized subunits within a cell with a specific function, and we'll be going through a heap today. And they can be membrane bound or non-membrane bound. The cytosol is the gel-like substance that organelles are suspended in, and that is a part of the cytoplasm. And then we've got inclusions as well, not really high yield, but thought I'd add it in. So these are metabolically inactive sort of cell products, such as carbohydrates, proteins, lipids, et cetera, et cetera. So they're pretty much random cell products that float around. Yeah. The next slide is a just list of important-ish organelles. Like I've said, not a lot of what I will be going through today will be assessed. So that's why it's not that important. But yeah, this is just a list for your knowledge that this is some of the organelles I'll be running through. Yep, next. So the first one is the cell or plasma membrane. This is pretty important because it comes up in semester 2 as well. And the thing to understand here is that the plasma membrane surrounds the cell. It is a phospholipid bilayer. So they have 2 sheets of phospholipids, and you have the non-polar tails facing inwards and the polar groups facing outwards. What this creates is a hydrophobic environment inside the membrane and a hydrophilic environment on the outside. Next, so within that membrane, there are also a few other things. So you have your phospholipids, you have cholesterol, which provides rigidity to the cell, you have proteins, and you also have carbohydrates. And in the proteins, they serve 2 functions. One is communication and the other is transport. And again, these are pretty important things because they will come up again in semester 2 as well. So yeah, next. So really important is that we understand the nucleus. The main thing to take away here that human cells are diploid, so we contain 23 pairs of chromosomes, and we have 46 chromosomes in total. And you have a diagram on the right as well. So in the nucleus there, you can see we have chromatin, and it's important to sort of understand the difference between chromatin and chromosomes, as I think someone else in our lecture will be talking about that in cell division. The nucleolus, the main thing that I took away from this last year, and the only thing I'd recommend you sort of know, is that it creates ribosomes. That's its main function. And also, if you were given a diagram, kind of just know how to distinguish between a nucleus and a nucleolus. So the main thing is that the nucleus is bounded by the nuclear envelope, whereas the nucleolus has no limiting membrane. So the next one is the mitochondria. So it is the powerhouse of the cell, like the name suggests. And what are some of the main things you should know? It has a double membrane, and it also, something else that's cool is that there are originally bacteria, which were integrated within cells by phagocytosis. And another small point is that we inherit our mitochondria from our mothers. And the next organelle I'll be talking about is the Golgi apparatus. And why is it important? Because we have the transportation of wastes and products is done by the Golgi. And a small point is that products are packaged at the cis phase and then released from the trans phase. And yeah, next slide. So ribosomes. This is a question that comes up pretty much every year, and it's that which of the following is not an organelle, or is not considered an organelle, and it is ribosomes. That's the correct answer, and that's because it's not membrane bound, so we don't consider it as an organelle. I remember getting that incorrect in my practice exams before the end of the year exam, so keep that in mind. And they're important for protein synthesis, as you'll probably learn in week 2 in the central dogma lectures that you cover, but nothing that important at this stage. Then you have your rough endoplasmic reticulum, or RER. Why is it called rough? Because it has ribosomes embedded into it. It's the site of protein synthesis, or the translation phase. And when sufficient amounts of protein are produced, they're packaged into vesicles that bud up from the endoplasmic reticulum and then go to the Golgi body. And it's often found directly around the nucleus. So again, I think you might have had a quiz for your cells 1 lecture, and I think in the quiz they have a diagram of a cell. So yeah, this could help knowing that it's near the nucleus. Then you have your smooth endoplasmic reticulum, or SER. Why is it called smooth? Because it's like the rough ER, but it has no ribosomes, and it's also continuous with the rough ER. And another point that might be important is that it's slightly different in structure. It's more tubular in how it looks. If you see in the diagram on the right, it's more tubular, whereas the rough ER is a lot flatter. Then, not too long to go, we have the difference between prokaryotes and eukaryotes. This, again, I just thought I'd put some of the differences and in a table. What is important? I guess it's the first sort of row that we have there, that in prokaryotes, there's no membrane-bound organelles. Eukaryotes, they're membrane-bound. It contains membrane-bound organelles. And the sort of the last row is important as well, that prokaryotes have pili and fimbriae, and eukaryotes have cilia, which Yufei will cover. And very last is, again, like I said, it's sort of important to maybe get a summary, put everything together. This is a typical animal cell. And as you can probably tell, we've covered most of the organelles, or the organs here, that are important. And the cytoskeleton, which is covered in cells 2, is what Yufei will be covering, and she'll start now.
All right. Yeah, just a reminder, if you have any questions, feel free to put them in the chat. Seeing some amazing questions coming through. Yeah, you guys are so knowledgeable. Yeah, and if you're feeling a bit tired, feel free to stand up, stretch a little bit, because It's been half an hour. But yeah, let's move on to cells 2. So this is your second lecture of cells. Yeah, next slide. So just a slide about the plasma membrane, which is the cell membrane. So really not too much you have to know about this, but just know that the structure is a phospholipid bilayer, and on the membrane, it's not just like a membrane with nothing on it. It has proteins, cholesterol, carbohydrates, and other things embedded on it, and all of these things have its functions, which we'll cover next slide. And the plasma membrane is selectively permeable. The previous slide. Yeah, it is selectively permeable. So what that means is some things can go in, but like not everything. So because it is a cell membrane, so like there are things that can go into the cell, for example, like oxygen and carbon dioxide and some like ions, some nonpolar molecules. But like there are other things that cannot enter the cell. For example, like water actually can't go in. Yeah, and the cell membrane is really thin, so it is only visible under electron microscope, not light microscope, because the light microscope doesn't have enough what is it called? Magnification. Yes, it doesn't have enough magnification to see the very thin cell membrane. And just as a side point, so like two general features of biological membrane. So like one is it is asymmetrical, so it is not the same on its outer on this like the outer side and the inner side. And like there are some proteins that are like there are some like proteins or like carbohydrates that are only on the outer side. For example, the carbohydrates are actually like only on the outer side of the cell. And also it is a dynamic and fluid structure. So what that means is actually like the things on the cell membrane, they can move around. They're not fixed. They're not just like fixed there. They can move around. So like the proteins, the cholesterols, they could kind of be at different locations in the membrane. Yeah, okay, next slide. Next slide. Yeah, so continuing on to what the lipids and the proteins do. So the lipids are generally the things that contribute to the structure of the cell membrane, while the proteins are responsible for its specific membrane functions. So some of these specific functions are transportation. So they could transport specific molecules across the membrane. So that ties into the selective permeability of the cell membrane. So basically the proteins control what can go in and what can't go in. And also the proteins can act as receptors for chemical signals. For example, like neuroreceptors and things like that. And the functions of the carbohydrates. So like the only thing you need to know is they're quite important in cell recognition. So like differentiate different types of cells. For example, in like for immune cells and things like that. And for the functions of lipids, so because they contribute to the structures, so they could serve as a selective barrier. So basically, like it's the barrier of the cell. Yeah, and also they affect the structure, the fluidity, and the permeability of the cell. Okay, moving on to next slide. So now we're moving on to the cytoskeleton. So the cytoskeleton is basically, so they're not organelles, but they are like basically filaments. So like protein filaments inside the cell. And what they do is they, so they have some functions. For example, they could maintain the cell shape. They could move organelles within the cell, and they could control cell movement. And oh, my laptop's gonna die. Give me a second. So I need to give me a second. I need to assign someone else host, and I'm gonna present from Ash's laptop. Is that okay? Sorry. 1, 2, 3. So right. I can't hear anything.
can't hear anything. Can I not use this? Okay. Okay. Can everyone hear me? Can someone say something just so that I can check this is working?
I can hear.
Yeah, all good, Yufei.
Can't hear anyone. Yeah. Okay. So yeah, basically, we're up to the functions of this cytoskeleton. So it could maintain cell shape. So they're quite important in like what the kind of the shape of the cell, and also like it could move the organelles within the cell. It could contribute to like cell movement, for example. Like, yeah, we're going to touch on that in the next few slides as well. And they have also like they contribute to certain specialized cell functions, like in meiosis and mitosis. So there are three types of cytoskeleton. We have microtubules, microfilaments, and intermediate filaments. So, wait. Oh, okay, present. Sorry, I might be a boomer, I might not be. Okay, so here's basically a table like comparing the three different types of cytoskeletons. You don't have to know too much about this, just know, just know there are three types of cytoskeletons. Okay. So first, we're going to talk about the microtubules. So basically, they are hollow tubes, and they are basically tubes made by like the protein subunit, which is called tubulin. And like you could see in the image, they basically look like that. You don't need to know any more than that. Basically, like you just know like it looks like that. That's it. And like so what else is important is like how does it assemble and the assembly. So what that means is so how does it make itself longer or shorter so that it could, for example, reach an organelle. So like that's done by polarization or depolarization of the tubulin dimers. So like what it does is basically like it adds tubulin dimers on itself, or like if it wants to like become shorter, it just like get rid of those subunits, which is pretty cool. And continue on, so like some functions of microtubules, maintain cell shape, cell motility. So that example of that is cilia and flagella, and also they're important in mitosis and meiosis. So we're going to talk about some examples of microtubules. So first one would be flagella. So that is, so a flagella is basically like a, it's like a structure on the cell, and that's which is mostly composed of microtubules. So it is often found on bacteria and also on sperm as well. So it helps the cell to travel in the host, or in the like person or whatever structure the thing is in. And the it has like for the flagella, and it has like this special kind of structure. Basically, the microtubules make itself in a structure that we call 9-2. So you just need to like remember 9-2. Pretty much that's the extent of knowledge that's required of you. But basically, if you want to know more, just like look at the picture. That's what it looks like. So it's like you have two like microtubules in the middle, and you have like kind of 9 pairs of it, like surrounding the center. Yeah, cilia basically has the same structure as flagella, but cilia's function is different. So like what cilia does, it helps. So you can find cilia in the lungs, and not the lungs. You can find cilia in the airway, and they basically help to move things like mucus along the airway. It helps it to like move the mucus out of, say, like our trachea, or like basically our airway. And the most you need to know is like it also has the 9+2 structure, and Yeah. Okay, moving on to centrioles. So that has basically all you need to know about this is it has it also is structured. So like it has it's formed by non-microtubule triplets and it's what's called a pinwheel arrangement. Yeah, and what is it? So this is involved in spindle formation during mitosis, which will be covered later in the session as well. All right, moving on to microfilament, which is the second type of cytoskeleton. So its structure is basically has two intertwined strands, and its main function is so like I think the reason why you need to know this is so like in the second semester you're going to learn about muscle contraction, and the microfilaments are quite important in that, and also. Microfilaments are also kind of contributing to plasma membrane activity, such as endocytosis, exocytosis. So basically it changes the shape of the cell membrane. And cytokinesis, you might have learned about this, you might have not. So like that's basically in a step in mitosis. And also it kind of helps with the cell shape. So they contribute to the cell shape. And an example of this is microvilli. So basically, don't confuse the microvilli with villi. So villi are like basically is composed of multiple cells and it could be found in your intestines. But microvilli is basically a part of the cell that's made up of microfilaments. Moving on, so basically this is more about actin and myosin. So we just included this slide here so that you kind of have a basic knowledge about like, so like the microfilaments are made up of actin and it's associated with actin, it's made up of actin and myosin, but you don't have to know too much about this. This is just like in preparation for for learning about muscle contraction. Yeah, so moving on to the last type of cytoskeleton, intermediate filament. To be honest, I think this is really not too important because we really haven't heard too much about intermediate filament after this lecture at all. So just know that it is a type of cytoskeleton and it also helps with cell shape and it has other functions as well. All right, so I'll hand it on to, so now we're going to talk about cell proliferation and death.
Yep, I think that's my slide. So we'll just wait for Ashwini to move on to the slide. All right, thank you. And yeah, these are just the main lecture outcomes, which you can look through in your own time. Next slide. All right, okay, so the cell cycle. I'll be going through the cell division mainly today. So basically the cell cycle is, the goal is to produce two genetically identical daughter cells from one precursor cell. So we want to split that initial cell into two and they're genetically identical, so they're the same. And cell proliferation is basically making new cells. So why do we need new cells? We might need them for the growth of a multicellular organism, or it might also be to replace damaged cells in the body. And so how do we achieve this? So first we duplicate the content inside the cell and then you physically divide the cell. There's two main steps and there'll be more involved, but I'll get to that later. And the types of cell proliferation, there's mitosis and meiosis, or meiosis, whichever way you want to say it. And we'll get to that a bit later as well. And basically, why is the cell cycle important? As you can see, cancer, if we have uncontrolled cell division, then it can possibly lead to cancer, which is not good. Yeah, next slide, please. All right, thank you. So for the cell cycle, as you can see on the right side, that's a basic overview of the cell cycle, and I'll try and go through the main things that you guys need to know. So for bacteria, the time for the cell cycle, it varies for each organism. So you don't really need to know the specific times, but just need to know that bacteria are much faster compared to yeast and mammalian cells. cells are usually the slowest. They take very long time. And then within the cell cycle, mainly there is the interphase, which is G1, S, G0, and G2, which you can see on the side there. And then on the last, you have mitosis and cytokinesis. Next slide, please. Yeah, so starting off with interphase. So there's 4 main phases. We usually say 3 because G0 is basically, it's where the non-dividing cells are. So for example, you have the nerve cells and they don't really ever leave that phase and they stay there and they don't replicate. Otherwise, you start off with the G1, so gap 1 phase. And in this phase, it's the longest period of the cycle. This is where the cell doubles in size. That's the main thing that you need to know. And then that's followed by the S phase. So the G1 phase basically prepares you for the S phase. And in the S phase, there's DNA replication inside the cells. And that's where you have 2 identical copies of each chromosome produced. And yes, we'll go on to the difference between chromatin and chromosome as well a bit later. And then that's followed by the G2 phase. So the G2 gap 2 phase, it's basically you preparing yourself, preparing the cell for mitosis, which is to come after that. So you're double checking the DNA for any errors, any replication errors, to make sure that you have a perfectly functional cell going into mitosis and you're not dividing any errors. Yeah, next slide, please. All right, so the cell cycle regulation, I believe this is quite important. So there's basically 2 key regulatory molecules that you need to know about. First of all, there's the CDKs, also called the cyclin-dependent kinases. And they're basically enzymes and they modify protein substrates that are involved in the cell cycle. And what helps these CDKs is cyclin. Cyclin is a protein and it basically binds to the CDK and it helps the CDK function as it needs to. So what these molecules do, they basically, as the title says, they regulate a cell's progression into the cell cycle. Yeah, next slide, please. All right, so I would say this slide, according to me, would be a bit low yield because I don't really think there were any questions that really focus on this, but yeah, I don't think you need to know the specifics, but it would be good to know the specific cyclins and CDKs if you can. Otherwise, don't stress too much. So basically, these are the specific regulators for each phase. So for the G1 to S phase, you have cyclin D. I won't go through it because but you can read through it in your own time. And yeah, and then you have the S phase and then G2 and N phase. But yeah, you can read through that in your own time. As I said, it's not that high yield. Next slide, please. All right, so we have mitosis. So mitosis is basically the second last step in the cell cycle. As I said, remember we had interphase, mitosis, and then cytokinesis. So in mitosis, we have 2 identical diploid daughter cells produced. So diploid means they have 2 sets of the chromosomes, and these are the phases for the process. So there's first, usually how I remember it is PMAT. So P for prophase, M for metaphase, A for anaphase, and T for telophase, and interphase comes before mitosis. So for cytokinesis, it's basically the division of the cytoplasm in the 2 cells. So it's basically cytoplasm dividing. That happens at the very end, but we'll focus on mitosis. Next slide, please. All right, so starting off with interphase. So interphase we covered actually already. That's basically where the chromosomes, they're in active transcriptional state, and the cell spends most of its time there. Like I remember when I said before, up to 24 hours there. And then early prophase. So early prophase, the main thing you need to know there is that that's where the chromatin starts to condense, or it condenses. And then in late prophase, the nuclear envelope disappears, the nucleoli disappear, and the spindle apparatus forms. As you can see in the image on the right up there, in late prophase, you can see those lines forming, and they are basically the spindles that will later attach to the chromosomes. So I would say this slide is very important to know. Like, it's important to know the key things in each step. All right, next slide, please. Okay, so metaphase. The spindle fibers, they grow, and then the chromosomes will attach to the middle of the cell, which is called the equatorial plate. It's important to note here that the chromosomes are most condensed in metaphase. That's an important thing to know. And that is why karyotypes are most likely performed in metaphase, because they're most condensed and most easy to see then. And I'll have a karyotype in the next slide so you can have a look. Then you have anaphase. Anaphase is basically where the centromeres divide and 2 chromosomes are made. So basically what happens here is that the chromosomes, which you have 2 sides to, they separate and you have individual chromosomes going to either side. Here the spindle fibers shorten and they pull the chromosomes to opposite ends. And then lastly, we have telophase. Telophase is basically where the chromosomes decondense into chromatin. So chromatin and chromosome, the difference between the two is that usually you have DNA. DNA is wrapped around things we called histone proteins. And once it's wrapped around, it forms a thin line called a chromatin. And then the chromatin then wraps around to make a chromosome. So basically a chromatin makes a chromosome. But yeah, it would be good to have a look at the image of it. And the nuclear envelope reassembles. Next slide, please. All right, so this is a normal karyotype. That's where you guys have to have a look. You can see the sex chromosomes at the bottom there, X and Y. So it's basically a female. Next slide, please. All right, so cell cycle inhibitors. These are also quite important. They will come up again, I believe, in semester 2 in terms of cancer when you'll study, in semester 1 as well. Yeah, you'll study cancer in semester 1, so these will come up. So cell cycle checkpoints, they are checkpoints they make sure that the cell is okay and it's ready to move on to the next stage. But if not, it can signal the cell death pathway. So cell death pathway, we'll come on to it later, but basically there's apoptosis and necrosis, basically telling the body to kill that cell because it's not functioning properly. So there's 2 main types of cell cycle inhibitors. So you have P53 and RB, which is also called retinoblastoma. So P53, it's named after its size, and basically it stops the cell from going from G1 to S phase. And why does it stop it? Maybe because there's some errors, or the cell it's not fully ready to move on to the next stage. So what P53 does here is that it basically activates the transcription of inhibitors, and they inhibit the action of CDK cyclin. And remember we said CDK cyclins help progress onto the next stage. So if they're stopped, then that means that the cell can't move from one stage to another. And most of the cancers, 50% plus cancers, are as a result of the malfunction of P53. So you have cells that are not fully functional going on to the next stage, which is not good. And then you have RB, and RB is the same thing. It also stops the cell from progressing, and it stops the progression from G1 to S phase. Next slide, please. So meiosis. So there's 2 main types of multicellular eukaryotes. So there's somatic cells. So that's any cell except for gametes, and then you have germ cells. So meiosis is the division for germ cells. Next slide, please. All right, so for meiosis, cell proliferation, it's a cell proliferation that produces gametes and sex cells. So the aim is to create gametes with only 1 copy of the organism's genetic information. So it's preparing it for sexual reproduction. This table is very helpful. It helps summarize both processes very easily. But basically, mitosis, you have 2 identical daughter cells produced, whereas in meiosis, you have 4 haploid daughter cells. The difference is meiosis, that they're haploid, so they only have 23 chromosomes. And the difference between the 2 is that meiosis has 2 cycles, basically double of mitosis, and it produces sex chromosomes. Next slide, please. All right, so for meiosis 1, these are the basic steps. It's very similar to mitosis, except for in metaphase, you have chromosome pairs that line up on the equator. And in anaphase 1, the difference is that homologous chromosomes move to opposite poles. So as you can see in the image on the side, the whole chromosome, the homologous chromosome, moves to each side. It's not split in the first stage. And then the rest of the steps are pretty much the same. And then moving on to the second phases, can you go to the next slide, please? Yep, so these are all the second phases. And as I said, it's basically mitosis twice. So here what we have is that in metaphase, the chromosomes once again line up, but then in anaphase 2, the sister chromatids, they separate and they're pulled towards either side. So that's similar to mitosis like we saw a bit earlier, but that's basically the difference between the two. There's two points of separation, and the first one you have whole chromosomes separate, and the second you have individual chromatids separating, so half of the chromosomes separating. And then the rest is quite the same, except for in cytokinesis, you have 4 haploid cell produced, as you can see on the side there. Next slide, please. All right, so genetic variation, this is actually very important as well. It can come up in your exams, and I think it's quite high yield. So it can happen by crossing over, independent assortment, or random fertilization. So for crossing over, it basically happens in prophase 1, and it's basically shuffling of the alleles. And in independent assortments, the chromosomes line up on the metaphase plate, and they line up randomly. That's the thing. So they can line up randomly, and that creates variation. And then you have random fertilization. Next slide, please. All right, so non-disjunction, it's basically when the chromosomes don't separate properly in meiosis. So non-disjunction in meiosis 1 is worse than in meiosis 2. And there are a list of conditions that can happen at the bottom. So you can have trisomy 21, 18, and 13, and Turner and Klinefelter. So that table there is very important. So make sure you know that which one corresponds to which. So trisomy basically means an extra chromosome. So it might be an extra chromosome 21, might be 18, or might be 13. It's basically an extra chromosome. And then Turner syndrome, you only have 1 X chromosome. And in Klinefelter, you have 2 X chromosomes and 1 Y chromosome. So that's basically the conditions that might result as a result of non-disjunction. Next slide, please. So this is orogenesis and spermatogenesis. So I don't think this is too high yield, but basically orogenesis is egg formation, and it's asymmetrical division. So basically you have 1 big egg produced and 3 polar bodies. You might be able to see on the side. Yep, so that's the ovum, which is the big egg, and that carries most of the nutrients. And then spermatogenesis, you have 4 equivalent sperm produced, as you can see on the side as well. And the maturation for the sperm is quite rapid compared to the eggs. And yeah, next slide, please. All right, so cell death, this is quite important as well. So there's 2 methods, there's apoptosis and necrosis. So apoptosis is programmed cell death, and necrosis is accidental. Next slide, please. I'll go a bit more into it. Yeah, so this is the steps for apoptosis. It's good to know the basic steps, but I think mainly it's important to know that the cell shrinks and the cytoskeleton gets broken down by caspases, and then the cytoplasm is dense and the chromatid condenses. Sorry about the background noise. And then basically you have the nuclear envelope begins to dissolve and it fragments, and then you form blebs, and the blebs are called apoptotic bodies, and they are then engulfed by macrophages. Next slide, please. All right, so apoptosis and necrosis. This table is very good. Please make sure you remember it. So basically, apoptosis is very clean. It's very well regulated division, where compared to necrosis, it's a bit all over the place. There's a lot of cell bursting and contents everywhere. So we like apoptosis because that means you're not affecting any other surrounding cells. There's no inflammation. Whereas necrosis is basically the cell bursts and it's damaging surrounding cells. And that often requires surgical removal because dead tissue is not phagocytosed. So that can be a bit difficult at times as well. All right, next slide, please. Yeah, and I think that's it. And I'll pass it over to Savannah, I believe.
Yes, hi guys, it's me again. So I'm gonna try and make this as interesting as possible. I guess one good thing about Zoom is that I can't tell if you're laughing at my attempts to be funny or not, so that's good. I'm going to try and put as many memes as I can in this, because I don't know if you noticed earlier, but I'm kind of the master of med memes. That's a bit of an exaggeration, but feel free to DM me, by the way, if you want any like memes about med content or anything. So yeah, let's just get into this. I think this is the most fun part of the med course, to be honest, at least first year. Next slide, please. So yeah, starting off with a meme, of course. So this is going to be your first lecture for tomorrow, by the way. So this is actually, for the first time, you're seeing content that you haven't seen before yet. So this is just a meme from last year. I think this was from the MedStart thing. So yeah, you're going to be a doctor. My3 is the bottom guy, and he's very interesting and very into whatever he's talking about. So I think you'll find him fun. Next slide, please. So what is health? Yeah, when the doctor tells you to take your medicine at 10, but you take it at 9 so you can surprise the virus. Okay, that's not actually health. That is health without the A. So just want to make that clarification there. But yeah, next slide, please. So actually, health with an A is defined by the World Health Organization as the state of complete physical, mental, and social well-being, and not merely the absence of death, disease, or infirmity. So that's a pretty longish definition. As far as I recall, we didn't have to like memorize this verbatim, but I mean, I think it's worthwhile just knowing what is kind of important from it. So the two key points here are, there's two problems with this definition. One is that complete health is not really achievable. Complete well-being is not really achievable because like there's always some aspect of our lives that's not perfectly in balance. And the other issue with this definition is that, well, who defines well-being? No pun intended by that. But yeah, so that's kind of very subjective, which is not definable. So there's some problems with this definition. All right, next slide. Okay, so data, which is a very important part of, well, analyzing health outcomes, basically, which is what I'll talk about in the next coming slides. So we have two types of data. There's primary data, which is the raw or processed data gathered from empirical studies. Empirical studies means studies based on observations. And comparable data is the primary data that is adjusted so that we can compare it between or among countries, because not that all data is, like, not that all countries have the same standards and have like the same, like there's always different factors that kind of affect how we analyze data. So comparable data is what kind of adjust for those confounding factors. That's a term you'll learn next semester, confounding factors. So that's something that kind of, yeah, makes it comparable. All right, next slide. So the main concept from here is, from this whole lecture, I suppose, is the difference between inequity and inequality. So inequity is when you have unfair, unavoidable differences in healthcare access. But inequality is when you have uneven distribution of health or health resources. So for example, the disparity between the resources in rural Australia and urban Australia, that's called inequality. They're not equally or evenly distributed. And inequity is, well, in a sense, inequity overlaps with inequality, because in that sense, it is unfair that rural Australia doesn't have the same resources as the rest of Australia does. But that's kind of the bottom line. They do overlap, but there is that difference there. So next slide. And what do we mean by avoidable? So there's factors that can be changed, like income and location. And therefore, unavoidable inequalities would be something like, for example, height. We can't change that. And I'm going to open this up to you guys. Can you think of any more examples of unavoidable or avoidable inequalities? You can turn off your mics if you're willing to speak. Anyone? I'm going to peek otherwise, so you better volunteer.
Your first language?
Yeah, that's right, first language. Sorry, would that be avoidable or unavoidable?
Unavoidable.
Yep, that's right. The language you're born with, that is unavoidable. Good. Anyone else?
Sort of genetic disease that you're born with.
Yeah, that would be sorry, would that be unavoidable or
unavoidable? Yeah.
Unavoidable. Yep, that's right. Just say that. Just
yeah. Okay. Right.
But yeah. And one more, just to see what's going on.
Your race.
Your race. That's right. That's good. That would be.
Unavoidable.
Unavoidable. Yep, that's right. It's pretty straightforward. So next slide. This is part of why I found this subject so fun. Anyway, next one. health indicators, so don't press yet. Yeah, so we have this meme dude. Oh yeah, I think I was gonna ask, wait, give me a second. Just want to check what he's on this. Oh yeah, so can someone define to me what health indicators are? Take a guess, just, it's probably what you think it is, to be honest.
Like the things the nurse takes when you first step into the hospital, like blood pressure, temperature, that kind of thing.
I like how you're already thinking like a real doctor. That is not quite what we're looking for here. So they're not we're not talking clinically. This is like on a global health kind of perspective. What do you think health indicators are?
Are they the measures which pretty much measure like the health status of the population?
Yeah, kind of. So yeah, so you can press next now. Yeah, okay. So health indicators indicate how healthy a population is. It's, yeah, literally, yeah, I do that. It's just exactly what it says. They're things that indicate how healthy a population is. They're like the statistics that we use. And this is where primary and comparable data come into play. They're kind of, and like we can kind of analyze the health indicators based on that data. So yeah, good job, guys. Next slide. So examples of health indicators are primarily DALYs, that's the main one you want to know. And there's also stuff like infant mortality, life expectancy, prevalence of diseases, conditions, admissions to hospitals, frequency of health risk factors. Now, can anyone think of any more by chance? It's okay if you can't, it's a little bit tough if you don't read statistics and stuff.
Can you say maternal mortality or how many children each woman has?
Yeah, that's a health indicator. I definitely think maternal mortality is a health indicator. Yeah. What else? Anything else? Sorry, big deal.
Things like health adjusted life expectancy.
Yes, that's another one. And I think that's it's very similar to what we're going to talk about with DALYs. So on to the next slide. That's a nice segue there. So DALYs stands for disability adjusted life years. And they're also known as YLDs, which are years lived with disability. Now, I really don't believe that you need to know how to calculate this because I didn't see any questions like this come up ever. But just so you know, DALYs are equal to the number of years of potential life lost due to being like more likely to die soon, plus the number of years of quality life lost due to disability. So life that you can't live to your fullest because you're disabled. Yeah, I think that's good. Next slide. Okay, so this is something that's in your lecture slides for tomorrow. Not lecture, I think it's the pre-lecture slides for tomorrow. This is not high yields, though. I'm just telling you this because it's there. So this is the physician's oath. I believe we will be swearing this when we are about to become doctors, for real. So they're basically just 13 points that revolve around like saying like, oh, the patient is the most important thing in our lives, patient's health and well-being. So we're not gonna compromise that for anything. Do not memorize this, by the way. You don't have to memorize it even when you're about to become a doctor. So just to know of, basically. All right, next slide. Just some key points from the oath, just so you're aware. So respect patient confidentiality even after they have died. This is something we'll be going into in more depth when we go into ethics in the coming weeks. Respect autonomy and dignity of a patient. Autonomy is also another important thing that we'll be covering in the coming weeks. Respect all human life, which I assume everyone will do because we're going to be good doctors. And I means you guys, you'll take care of yourselves and update your knowledge and skills for the sake of patient care. And you'll honor the medical profession and its noble traditions. So yeah, that's basically, you don't have to remember this, it's just stuff to know. This is kind of important, but I wouldn't say it's high yield either. So controlling an outbreak. So in case you don't know what an outbreak is, it's basically when disease, goes loose in a particular population. In case you couldn't tell, doctors are really essential in controlling outbreaks. So we start by testing or investigating the patient. Unfortunately, I can't think of any examples right now of what an outbreak could be, but maybe you can think of your own. So test or investigate the patient. And then you would trace contacts to see like if anyone else got got it from the patient, and then you'll offer treatment. For example, quarantine is a valid treatment. Not sure if you guys heard about that recently in the news. And you would report to a public health official if appropriate, while maintaining patient confidentiality. Confidentiality is really important when you're a doctor, so just keep that in mind. You'll always hear about it. And after this, public health official will gather population data. Here's where data comes into handy, and inform government officials who inform the media, and that's why we get stuff to news. Okay, so while I say this process, it's not like gonna work 100% of the time. Otherwise, otherwise outbreaks would be gone in like a week maximum, but they don't. So the above method doesn't work so well in reality because there's issues like underfunding of health services, not enough clinics, and too few or overworked staff, and patient morbidity and mortality increase. Yeah, so that's kind of why we still have outbreaks happening. Yeah, okay, I think that's it for this slide. Oh yeah, that's all, folks, and I'll probably put that every time I finish my section in the future. And on to the final presenter, who is...
Hey guys, so I'm covering cells and body fluids. Our first physiology lecture for the year. How exciting. Okay, next slide, please. Right, so I'll begin with some chemistry from last year. So concentration, we all know that it's mole over liter. I hope we all know that it's mole over liter. And so for H2O, remember that 1 liter of water is the same as 1 kilo of water. So the calculation would be 1,000 grams over the mole mass, and then you'd get 55.5. And that basically means 55.5 moles of water are present in a liter of water. And so a few other terms that you should remember are solutes, which is the substance that is being dissolved, and then solvents, which are the substance that the dissolving happens in. So, for example, water. Okay, next slide, please. Okay, some other new terms that you might not know of. So intracellular fluid is the fluid that's inside the cell, and this makes up 2/3 of water in the body. Extracellular fluid is fluid outside the cell, and that's 1/3 of the water in the body. So osmolarity is the number of solute molecules over the volume of the solvent. So pay attention that it's the number, not the amount or anything like that. So all you have to do is just add up the number of molecules, and if there's different types of particles, you just add them up. Osmolality is the same thing, except instead of volume of solvent, it's kilograms of solvent. An ideal solution is when a solution is dilute enough so that the solute particles don't interact with each other. And effective impermeability is when the solute diffuses through the membrane, but it's immediately pumped back out. So basically it's impermeable. And so with the sodium potassium pump, the potassium leaves the cell but is immediately pumped in, and so for sodium it's the opposite. It enters the cell and it's immediately pumped back out. So that's just an example of effective impermeability. And so next slide, please. Okay, and so this is just a little bit of a diagram about how the fluid in our body is split up. So I was saying before about the intracellular fluid and the extracellular fluid, and that's divided up even more with intravascular fluid, which is the fluid within blood vessels, interstitial fluid, which is fluid between cells, and transcellular fluid, which is fluid found in the gut, in the eye, and cerebrospinal fluid as well. Next slide, please. And so with blood, when we say blood, we mean collectively both the liquid and cells found in blood vessels. Plasma, on the other hand, is just the fluid that remains when the cells are removed. And so that's like in the image down below. And serum is fluid left over when the plasma has been allowed to clot. So plasma includes clotting factors like fibrinogen, whereas serum doesn't. Next slide, please. So this is just a table about where different ions are found the most. So sodium is on the outside. That's why when I was talking about the sodium potassium pump, sodium is trying to come inside the cell, but it's immediately pumped back because of that pump. So this isn't exactly, you don't have to remember the numbers, but just know that there's more sodium outside than inside, and there's more potassium inside than outside. Okay, so moving on to diffusion. Next slide, please. Okay, diffusion is basically when cells, when particles are moving down a concentration gradient, like this cat over here. And so how fast diffusion occurs depends on temperature and some other factors as well. And if there's a membrane that is permeable with the solute and the solvent on both sides, they would have the same concentration after diffusion, but not necessarily the same number of particles. So it's all about the concentration. Osmosis is different from diffusion because it refers to a situation where the membrane is permeable to the solvent, so for example water, but not the solute, so what's being dissolved in the water. And we'd refer to this membrane as semi-permeable. But it still involves the principle of diffusion where particles are going down their diffusion, down the concentration gradient. So instead of the solute moving to a different area, it's the solvent. So the solvent is moving to the area with a high concentration of the solute. And so it's really important with these questions to know the type of membrane and whether or not it's permeable to the solute. So that's a pretty important point. I think I remember it being a question in an exam. And so in this image, you can see that the right side is more concentrated. And so the water will go to the right side. So that's why you can see a difference in the level of water. And so if you want to think about what's happening in the body, you have to imagine that the left side is really, really large, so that when water is moving into the right side, you don't really see a difference, unlike what you see in this beaker. Okay, next slide, please. Okay, osmotic pressure is the pressure that occurs when water is moving across. And this is because of the difference in the non-diffusibles, so what can't make it across the membrane. And so again, it depends on the membrane type, solute, and the solvent. And so how you would measure it is you would think about it as the pressure required to stop the water moving to the side that is that has the high concentration of non-diffusible solutes. So just like the arrow is what I'm referring to here. And effective osmoles, just another term, is non-diffusible solutes that help generate osmotic pressure. So these are the solutes that can't move across to through the membrane to even out the concentration. So therefore, the water has to move to the area where these effective osmoles are. And osmotic pressure will change depending on that concentration of non-diffusibles. And if a membrane was permeable to everything, then the solute would also be able to move across, and therefore there wouldn't be osmosis. So remember, osmosis is when there's non-diffusible solutes. And the difference between osmolarity and osmotic pressure is that osmolarity only involves the number of solute particles in a solvent, not the type of solute or the membrane, unlike osmotic pressure, which does involve two solutions divided by a membrane and non-diffusible solutes. Okay, so just a lot of terms, but here's another one, tonicity. So it's talking about the non-diffusibles. So hypertonic is a solution, it refers to a solution that's outside a cell that has a lower concentration of solutes than inside the cell. So if we're saying there's a red blood cell and the solution outside has less solutes than there are solutes inside the red blood cell, there would be movement of that outside solution into the red blood cell. I'll go into it a bit better later on in the later slides. So bear with me. Isotonics refers to a solution that's outside the cell that has the same number of same concentration of solutes as inside the cell, so there's no net movement. And then hypertonic is the solution outside the cell has a higher concentration of solutes than inside the cell. So there's going to be movement of water or fluid from inside the cell to the outside solution. Okay, next slide, please. So basically in the exams, I'll ask you all these scenarios regarding red blood cells, and these are the 4 solutions that they might ask you about. So first of all, the osmolarity of red blood cell, that's 300 milliosmoles per liter. And so its contents are potassium and anions, and they're non-diffusible. So these are non-diffusible osmoles. And then, so if we're thinking about it in the body, you have to imagine a red blood cell and there's a huge pool of solution. So there's no, any movement of water doesn't cause like a change to the solvent. So you wouldn't see any decrease in the outside solution level. So with water, which is a possible scenario that might give you, it's not applicable clinically because your patient will die. You'll find out why soon. And then there's 1.8% urea, 5% dextrose, and then normal saline as well. And urea, dextrose, and saline are all isoosmolar to the red blood cell. So as you can see, 300, 278, 308, that's pretty close to 300 of the red blood cell, which is pretty important in the next scenario, which I'll show you. Next slide, please. Okay, so if we're putting a red blood cell in water, we know that the initial osmolarity of the red blood cell is 300 because that's what we've been told. And then water will have 0. Tonicity, so that's the amount of the solute that won't be able to cross the membrane, so that's 300. And then the water has none. And so obviously there's nothing that's going to be able to go through the membrane. So the place of higher concentration is inside the red blood cell. So the water is hypertonic to the red blood cell, that therefore the water will move into the cell, which will cause the cell to burst. So that's called hemolysis. So that's why it's not used clinically, because your patient will die. Next slide, sorry. So the next scenario is red blood cell in urea. And this is also not used clinically, but both the red blood cell and urea have the same initial osmolarity, but the red blood cell has 300 tonicity, so there's the solutes that won't be going through the membrane, but urea does have diffusibles. So that means there will be something going through the membrane. So urea will diffuse into the red blood cell, and so there's a change to the osmolarity. And because there's something moving into the red blood cell, the cell will undergo lysis, and your patient will also die. So we don't use water and we don't use urea either. But they're just examples that they want to use to make you think in exams. Next slide, please. So normal saline we do use clinically. So 300, so the same initial osmolarity and the same tonicity as well. So there's no movement of water in or out. And it expands the extracellular fluid. And so it can be used in situations such as dehydration, blood loss, or other fluid loss. Next slide, please. Okay, so dextrose, this is slightly different because dextrose is only an effective osmol for a while, whereas urea and water are always not effective osmols, and normal saline always is. So this means that whilst it's there is no net movement of fluid into the cell or out of the cell, over time there is going to be movement of the cell because the dextrose will become CO2 and H2O, and because there's water, there is going to be movement of fluid, but there won't be that result of hemolysis. So you'd use this for dehydrated patients, and it would be used to expand total body water. And that's the last scenario. So hopefully that all makes sense.
All right, if anyone's got any more questions, or if there's any questions that we haven't been able to answer that you guys put into the chat, we will get back to you. We're also going to put up an anonymous like Google Forms sort of thing, so you guys can send in your questions and we will answer them next time. We know that we've kind of run over time, but next week definitely won't be this long because we will be sort of tailoring it to the stuff that you guys want to be covered, and we obviously won't have to do introductions and all of that again. If anyone else has any burning questions that they need to ask us right now, or if any of the other members want to say something before we log off.
Maybe just thanks for coming down. I think Sav was probably about to say the same thing. Yeah, thanks for coming down. Wishing you guys the best of luck this year. And yeah, hope we can help out in the journey that is Year 1 Med.
Yeah, and I just want to say, you guys are all amazing, and looks like you've all got really good questions, and I feel like you're a lot more keen than definitely I was last year at this time. I was quite dead at this time actually, last year. But yeah, I'm sure all of you will do really well, and we'd love to see you at our next session next week this time. So yeah, thank you for coming.
Yep, just really quickly, we will be putting, sorry, Sav.
Oh, okay, it's okay.
I was just going to say we will be putting up the links to everything, so our YouTube account and the Google Drive and everything will be going up on our FB page for anyone who missed the link in the chat before. Sorry, Sav, go ahead.
Yeah, okay, sorry. I just want to say thank you guys so much for coming to our pilot session of PSParachute. I hope we were as entertaining as possible. So next week we won't be doing introductions, of course, so really sorry about going way over time, but yeah, we should be able to be more concise next week onwards. And stay tuned for more interesting stuff coming up. Oh, next week we'll be having a Kahoot at the start of the week, so definitely come for that awesome lobby music, if nothing else. Yeah, see you guys. Thank you for coming.
Before ending, can I ask